scanning of by using the scanner for, which is densitometric technique. And if there is needed of derivation, you can go for derivation, and again image documentation and scanning part. And after the scanning, you can go for the bioactivity analysis like DPPH or any other microbial studies, and then EDA effect directed analysis, or if MS was or IR-NMR, you can go. Now CAMAG HPTLC for Squeezy and Research Labs. So HPTLC system comprising series of independent instrument and controlled by the only one software. So there is a different modules for each applications. Like for application there is a ATS4, then for development there is a ADC. So there is a different models of HPTLC, and up to the sixth analysts can use one HPTLC simultaneously for different QC and R&D samples. So and it is easily upgradable. Then database oriented software was here. So each analyst work is password protected and easy to track, search, correlate, and compare the reports and data. So multi-level security. Then minimal use of solvent. That means for 15 to 20 samples, you can use 30 ml is sufficient for the development. So and GR grade solvent you can use very economical and little waste was produced. Analysis on demand, if you see, so there is no disturbance to other users, and you will get the reports in 60 to 90 minutes. And the most important is the trustworthy Ancrom support since 1978. If you have any queries, doubt, any help you want from the Ancrom laboratory, we are there to help you. So Ancram has 40 plus years experience, and we give the trustworthy support to our customers. Now HPTLC regulatory acceptance. So USP chapter 203, it is for the HPTLC methodology and fingerprint identification of botanical origins. Then USP chapter 621 for the scanning of densitometry for quantification. USP chapter 202 for identification of the fixed oil. USP chapter 2251 for synthetic API in herbals, what HPTLC MS. USP chapter 1064 where parameters selection explanation. Then European Pharmacopoeia, Chinese Pharmacopoeia, WHO, they accepted this HPTLC method for the analysis. Now, these are the essential requirements for doing the HPTLC analysis according to the USP chapter. So there is fixed sample application positions and band length. Then you should go for 20 by 10 plate. So this all the essential parameters was required to do the HPTLC analysis. You can fulfill this by using the CAMAG HPTLC system. Like first application, so by using the ATS4 or Linomat 5, you can do this analysis. Then for the chamber saturation and humidity 33%, you can get this by using the ADC2. For development and derivation, derivatizer was there. Photo documentation 254, 366 white light you will get by using the visualizer. So this all the essential requirements for the quality work of HPTLC you can fulfill or you can achieve by using the HPTLC. Now the parameters which are decided in USP chapter 1064 is after the several experiments done, and after that they decided keep for which parameters and which parameters should be there for doing analysis. First, the you can see the application pattern, then the band length and the difference between the spot and band. Then the band length should be 5 mm, 6 mm, 8 mm, like that they did. And after that they decided the 8 mm will be the good band length. Then distance to solvent level, like developing distance also they did for 3 centimeter, 4 centimeter, 5, 6, 7 centimeter. And after that they decided 7 centimeter will give you the good resolution, and it will almost cover the two-third of the area of plate. Like that these parameters was decided, and that all explained in the USP chapter 1064. You can see the one sample application plate like the dummy plate. You can see this image. Consider this is 20 by 10 plate. So on 20 by 10 plate you can do the 15 number of bands. The x distance you have to keep the 20 mm, and the y distance will be the 8 mm. the band length on the plate is the 8 mm. You can do 15 number of tracks, and till what distance you have to develop the 70 mm. So this is one sample application of the 20 by 10 plate. Now, CAMAG HPTLC regulatory compliance, it is 21 CFR and Rule 11 compliant. You will get the log data. Good laboratory practices, CGLP and CGMP compliant, IQ, OQ, PQ compliant. There is all the data is securated, passwords protected and encrypted, digital signatures up to 5 level, and user access control up to the 7 levels. Then full audit trial for analysis and system. Now most of these are regulatory should be compliance in pharma. So these are parameters are in there in the software. Now, HPTLC sample preparation, how you can do the sample. Suppose you have powder sample, you weigh 100 mg in 1 ml of suitable solvent like methanol, ethanol, where it will be extracted and completely dissolved. You can go for the sonication for 15 minutes and then centrifuge at 5000 rpm for 3 to 5 minutes and take a clear supernatant solution for the analysis. So very easy, simple sample preparation technique were economical and fast. Now we'll start with the application with the each module applications and its features. First is the sample applicator. So Linomat 5 and ATS4, then robotic applicator pro. So these three are the applicators. By using these three, you can do the application on the plate. Now the difference between the Linomat 5 and ATS4 is only that one is the fully automated and one is the semi-automated. In ATS4, you have to prepare the sample, fill it in the vials, and keep in vial tray. Give command from the software; it will automatically take the sample and go for application. In Linomat 5, manually you have to rinse the syringe, manually you have to fill with the sample, and manually load and press the enter button to start the application. So this is the only difference. Otherwise, the principle for both the technique is the same. You will get a very precise and accurate application. Both instruments are regulatory norms met and integrated into the HPTLC system. Now you can see these are the some application techniques unique to HPTLC. First is the normal technique where quantitative, where you can apply sample and standard, you can do comparative, and you can plot the linearity graph and go for quantification. Second one is the in-situ cleanup technique. Now this mostly technique we used when there is a fatty materials or the samples which you have the oils or fats. So that time you need to extract that fatty matrix from the samples. That time you do application at 110 mm onto the plate, and then you develop that plate in ether, and we all know the ether will extract the fatty fats from the sample. And then you cut that plate at 120 mm from the bottom and turn 180-degree, and again you develop that plate from that side, so which was not possible in LC. Then micro preparative isolation study you can do, and superimpose, that means spiking study also you can do, or do application by using the applicators. Now next is the development. You can see these are the development model. First is the manual technique, twin trough chamber, which is TTC chamber. Like this TTC, you will get the three tanks, 10 by 10, 20 by 10, and 20 by 20. So depends on the number of samples you have. You will select your plate and the tank for the application and development. Very easy, simple technique to develop, and it will require very less amount of the solvent. Like 10 by 10 chamber required only 10 ml of mobile phase, 20 by 10 chamber you required only 20 ml of mobile phase, and 20 by 20 chamber required only 35 ml of the mobile phase. Then ADC2, automatic developing chamber. When you want to find out the reproducible result, or you want to do any literature or research work and you want to publish, you definitely go for the ADC technique, because it will give you a very reproducible results and accurate precisions wise separation of bands. And third is the AMD2. AMD is the automobile multiple development models. This is mostly used for the R&D or method development work. And there is a robotic development module Pro was there. Now mobile phase. very little consumption, as I told you, 20, 30 to highest is the 35 ml for the 15 samples. And any combination of the stationary phase, that is our plates and mobile phase, you can do. No preparation time, no degassing is needed for the sample. Very less time is required, almost in 1 hour you can complete your analysis. If suppose if any analysis goes wrong, you can throw that plate and take a new fresh plate, which is very cheaper as compared to the column, and you can do again the fresh analysis on the plate. Now, as I told you, there is a twin trough chamber TTC tank. So, like this TTC tank, look, there is a trough at the bottom that will divide your mobile phase into two parts when you tilt it to 45-degree, and that will divide your mobile phase equally to two parts, and you wait for the saturation. Now, at the one side, you have to keep the saturation paper, which is Wattman filter paper number one, and second side you can keep the plate for the development. And very less amount of solvent is needed, as I told you. 10 ml is required for the 10 by 10 tank, and on 10 by 10 tank you can do the 7 number of bands. So on one plate 7 bands you can do, and 10 by 10 10 ml is sufficient for the for development, and 20 by 10 you can do the 15 number of bands, and you just require 20 ml of mobile phase. Now the next is the ADC2, which is CAMAC Automatic Developing Chamber, fully USB compliant module, software control, fully automatic, and you will get the reproducible results. It will give you the humidity control. Near, as I CAMAC suggested to, you will get the 33% of the relative humidity if you use the saturated solution of the magnesium chloride hexahydrate. And then layer equilibrium, auto monitoring of solvent front. So in this module there is one laser light which is monitoring that development, and when it reaches to your desired developing distance, automatically that plate will take out from that chamber and it will automatically dry, and you can use that for the photo documentation. So fully automatic developing chamber. Like this, as I told you, there is a AMD2 also, automatic multiple development. So this is mostly used in the research work or R&D work. So suppose here this AMD2, there is a 5 bottles, you can fill it with the different solvent like polar to non-polar medium polar solvent, and then you can decide key till what distance I have to develop with which solvent. So you can do the multiple development in the same direction with the different mobile fractions. Then after development, the next step is the photo documentation step. So for photo documentation step, this is the visualizer you can use. This visualizer having high definition CCD industrial camera, which will give the three illumination mode, HD photos, a white light photo, then UV 254 nanometer, and 366 is for the fluorescence. So here you can see the how you will get the photos from the visualizer. Then in the software there is a one comparisons tool is there. You can compare the data with the different plates or the samples or different days analysis labs or any different species or the different analysis. So at a time you can collect that data and you can do the comparison also, and with that you can go for any conclusion key if any extra band was observed or any degradation is there, like that. So this is one beautiful feature in the software comparison. Then HPTLC quantitative evaluation. Now scanner is the very much important part of HPTLC. It is acts as a detectors. If you have scanner, you will get the accurate RF value. You will get the area for the peaks. Uh, then you can plot the calibration graph. You can go for the quantification analysis. If any unknown band was there, you can take a spectra for that, and spectra will give you the lambda max value. So all this you can do by using the scanner. Now this scanner has three lamps for scanning. So one is deuterium for UV scanning, then mercury for fluorescence scanning, and tungsten lamp is for the white light. Now, this is scanning in absorbance mode. Suppose you keep the plate under the scanner. Then you select the lamp which you want to scan. That the lamp light will pass from the monochromator. It will reflect from the mirror, and it will go onto the plate, and it will convert that bands with the help of photo multiplier tube and into convert it into the peaks. So see here the chromatogram evaluation. Suppose this is your plate, and the at RA point one, this is the band. After the scanning, you will see the peak like this in the software. Then, as I told you, there is one function of the spectrum analysis. So you can take a spectra, and for the different samples, there is a different type spectrum pattern was there. Like here, you can see the sulfur methoxyl at 260 nanometer, then trimethoprim as 210 nanometer, and dimetrazole for 318 nanometer. So you can see at a low UV level also it can detect wavelength also like 210. As I told you, it range have from 190 to 900 nanometer. Then if you take a spectra one time, it will give you the lambda max value, the identity of that band. But if you take the spectra three times, it will give you the purity of that sample. Now, you by using the scanner, you can do the multi-wavelength scanning also. So at a time, you can scan for different wavelengths also. Like here, the one example, 311 nanometer through 205 nanometer and 268 nanometer. So there is one option for the multi-wavelength scanning. Then quantification by HPTLC scanner 4. Now, why the scanner is needed? This is the one example. The TSC means tetrahydrocannabinoid cannabinoids, which is not visible in 254 nanometer where we took that image. But if when I go for the scanning, that time at 210 nanometer, it was detected. So there you can see the one green peak was observed. So when we take the photos, but it is in the UV, it is not visible. It is not like that key, it is not there onto the plate. But when you do the scanning, that time it was detected, and that is why the quantification of HPTLC scanner is required. Now the derivatizer. Now derivatizer is the technique which is patent. This derivatizer technique from the CAMAC. Derivatizer means what? After sometimes after the development we took the photos, but in the UV white or fluorescence light we can't see any bands or the separation onto the plate. So that time we go for the derivatizing procedure. So we deep that plate or we spray that plate with the specific reagent, and that will give the colors to the band which are present or separated onto the plate, and that procedure is called as a derivatization, to giving colors to the band. Now, by using this scan derivatizer, you can do the spraying of that specific reagent like dragon drop or the anisaldehyde sulfuric acid reagent, vanillin as vanillin reagent. It works on the piezoelectric effect, and it will give a extremely fine spraying, fully reproducible results. And the benefit and the advanced feature is that it will require only the less amount, like 3 ml is reagent is needed for the whole 20 by 10 plate. And after the finish all the spraying, the fumes will be collected. There is one bottle behind which containing the ethanol and water mixture inside that. Hello, hello, Farida sir, Farida
Yes,
sir,
sir. Your screen
Farida
is
sir.
not visible again.
I can hear the echo. 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 Just I will again join. 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 Hello. My
Yes,
screen
sir.
is
It's
visible.
visible now.
Okay. Okay. Thank you, ma'am.
Yes, sir.
Okay.
Yes.
Hmm. Now, this one is the spraying module, which is of Kamak pattern technique was derivatizer, and now this is the second module. It is immersion device. Now, this technique is a dipping technique. So there is one chamber come with this module. You have to fill that chamber with that reagent. Here you require nearby 180 to 200 ml of the reagent. Then there is a holder, plate holder. You can attach there your plate and just press the red button. It will go and deep in that tank. So this is the derivatization immersion device. So these two techniques used for the derivatization, and many times after the derivatization you require to heat the plates. So for that, this is the Tamac TLC plate heater. So it will give you the full uniform heating all over the plate, and the range of this temperature range of this plate heater is from 25 degrees Celsius to 200 degrees Celsius, and the surface of this plate heater is resistant to many of the solvents, acids, or the different solvents, and easy to clean. Now the next is the HPTLC fingerprints. As we know, every human having their unique fingerprint like that. This is mostly in the herbal technique. Fingerprint technique was useful because every plant also having their unique fingerprints. Like suppose turmeric powder was there. So in that turmeric powder, the curcuminoids or the dimethoxy or bidimethoxy curcumin bands was there. So on that basis, we can differentiate and we can identify that turmeric powder. Like that, this is the fingerprint technique. So the definition is that from the USP, it is the electronic image of the visual HPTLC chromatogram, and on basis of the RF color and intensity of that bands, you can uh, identify and you can compare it with different standard or samples. So for comparing, there is some botanical reference materials also there, which was authentic standards. And with the fingerprintings, you'll get images in 254 like UV 366 and white light, and you can stack that data for your research work. Now here you can see also one HPTLC fingerprint is unique because here you can see the different species of the Piper class with the different like Piper nigrum, longum, retrofractum, cubeba, and each one having their different patterns. Like any in Piper cubeba, you can see there is one orange intense band was there, which was not present in other samples. So this is the main marker component, and on basis of that, you can differentiate and you can identify the different species of Piper. Then after the fingerprints, you can go for the different class of compound studies also. Like there is a 14 different class of compounds: alkaloids, phenol, tannins, beta principal, coumarins, steroids, sterol. So each class of compounds with by using the different reagent, derivatizing reagent, you will get these beautiful colorful bands for the class of compounds. Now the next is the HPTLC MS interface techniques. So by using this, you can elute the pure HPTLC fractions into the MS, and you will get the molecular mass for that unknown band. So very useful technique for confirmation and characterization. Mostly used in pharma, in R&D, even in the foods. It is suitable for any MSMS or HRMS or MS, and it will only transfer the selected and pure fractions for the MS. Now, how it is works. So you will keep this interface between the HPLC pump and mass spectrometer. So the outlet of the HPLC pump will be connected to the inlet of this HPTLC interface, and the outlet of the interface will connected to the inlet of the mass spectrometer. So from that HPLC pump will provide the solvent. It will dissolve that specific band, and that will elute and it will transfer to the MS, and within 10 to 15 seconds you'll get the fractions, and you will get the molecular mass for that unknown band. So advantage of this HPTLC MS hyphenation is very simple to use. There is a 3-minute cycle per MS and only pure fractions eluted to MS. Cost effective analysis up to the 100 ms you can do per day for selected fraction of interest, and it will increase the existing LCMS or LCMSMS productivity of any make 10 times. This is the USP chapter where it is authorized adulteration of dietary supplements with the drug and drug analogs by HPTLC, MS, etc. This one is the one example of identification of API, tadalafil, in Ayurvedic sample. So it was detected and then further took the confirmation with the help of MS. We all know the tadalafil is the API, active pharmaceutical ingredient. It should be not present in the Ayurvedic sample. Then this is the Shimadzu and Ankram joint poster ASMS conference which was held in the USA. So this one is the poster where we characterized and isolated the quinones from the Tectona grandis leaves. This is bioactivity detector via bioluminescence, Camac bioluminizer. Now, these luminizers you can use to detect the toxicity in your sample. So, how you can detect that? This is not completely related to the HPTLC, but indirectly, the you have to follow some method of the HPTLC, like the application development of the plate, and after the development, here you have to deep that plate into the alivibrio fishery solvent, which is microorganisms, and you deep that plate and you keep it for some time or the some hours in the dark, and then you capture that plate image by using this luminizer, and it will show you the toxic bands or any like black color bands if you observed on your plate. So that will show that there is some toxicity or some toxic material was present in your sample. Mostly we use the alivibrio fishery because it is very safe to handle, and when there is any healthy colonies of these bacteria in your sample, so they will alive onto the plates, they will emit the lights or bioluminescence, and they are very much sensitive to even minute level of toxicity, and that is why mostly we use the alivibrio fishery. This is the one fingerprint of the wastewater sample. Now, bioluminizer applications, so selective detection of biological activity in synthetic mixtures and even in natural product extracts. Then toxicity profiling of water you can use, toxic residues materials detection, even in the food, beverage, dietary supplements, then even in the soil also to find out the toxic residues, you can use this bioluminizer. Common HPTLC biases like bacterial and yeast assist. So as I told you, the alivibrio fishery for toxicity, like enzymatic or antioxidant you can do for DPPA study, for radical scavengers or antioxidant present is there, hill reaction, herbicides. So these are the common HPTLC bioassays. Now, HPTLC method development and validation. So, by using the HPTLC, if you want to do the method validation, it is very simple and rapid. You need few plates and 1 day. Validation takes nearby 3 to 5 days. New method development and validation cost is rupees 25 to 50,000 only. If you compare to other techniques, it is very cheap and possible to create and validate 10 to 15 methods per month on HPTLC. Now, according to the ICH-Q2R1 guideline, these are the HPTLC method validation parameters where we follow the specificity, then precision, intraday and intraday accuracy like recovery, the linearity, LOD, LOQ, limit of detection, limit of quantification, and ruggedness. Now, as I told you, there is the updation in HPTLC, and last year in 2021 there is a robotic HPTLC was launched from Camac. This is the Camac HPTLC Pro, which is fully automated sample analysis and evaluation system for routine control analysis. So these are the modules of this HPTLC Pro: applicator, development, advertisation, detection, and MS interface.
So they all are interconnected with each other. There is one conveyor belt which will pass the plate after the each application or each step was completed, like application was done. So automatically that plate will go for the next part development and next part derivatization detection, like this. So this is the automation.
It is like a relay race. So you can see here in this video how this new robotic HPTLC works. Here is one video key how actually it works. So till date only the two modules was launched, applicator and development. So with this video it is very easy to understand. Then now we'll start with the application areas where HPTLC is mostly used. So pharma, food and adulterations, botanicals like herbals, industrial analysis, reverse engineering, organic chemicals, reaction mixing, and the forensics. So first we'll start with the pharma applications. Now, this is the example one analysis was done in our lab. Quantification of glimepiride in 1 mg of tablet. So we know the glimepiride is the anti-diabetic drug, and we detected that in a UV light, and then we plot the linearity graph, and it is detected. So you can see how the graph you will get, how the results look like glimepiride 133.3 mg in 10 mg of the tablet. Then second example is the quantification of the apexaben and apexaben tablets. We know the apexaben as a anticoagulant or blood thinner tablet, which was used to prevent the blood clots and to prevent a stroke in people with the non-valvular atrial fibrillations. So it is detected in the fluorescence mode, and then the plotting the graph. After plotting the graph, we detected that key it is 96.2 μg was present in 100 μg of the tablet. Method development and quantification of miltefosine in tablet and capsules. The miltefosine is the antimicrobial drug capsule that was visible after the dermatization. So you can see this image of after dermatization. Then degradation profiling of cefixime and azithromycin studies. Now we know this both are the antibiotics drugs. So what the madam done in this study? They degraded in the different mediums like acid, base, oxidation, heat, photosensitive, like this. And for cefixime, it was observed in heat. The that sample or the tablet was degraded, and for azithromycin, it is degraded in the acidic medium. And this publication won this the international award of Euro 2000 at Berlin. And the force degradation studies is the integral part of our drug development programs, and for that the HPTLC is a very specially beneficial for this stability testing study, very easy and rapid technique. Then simultaneous analysis means for the FDC, fixed drug combinations, levodopa, carbidopa, and tocapin. So you can see this is the example. So fixed drug combinations are not regulated by pharmacopoeia, and therefore it is ideal for HPTLC analysis, which is 10 times cheaper as well as the faster as compared to the AC. And any FDC analysis method can be validated by using the
HPTLC. The next example is the HPTLC method for the ulipistral acetate in presence of its forced degradation products. So ulipistral is the emergency contraceptive tablet which was used for the to prevent the uterine fibroids like that. So degradation studies were done by the using HPTLC, and HPTLC MS is a simple, quick and low cost, and very visibility to the sample is also very helpful here. Then HPTLC
determination of the diclofenac sodium from the serum sample. So this method is very sensitive and reliable, and further studies by MS are possible. Cleaning validation by HPTLC. We all know in the pharma sectors, after the finish of each batch production, they need to go for the another. But before that, they clean the vessels, and after that, that solution or the solvent cleaning we can apply onto the plate, and after that we can detect it is fully cleaned or not. So the in the cleaning validation also we can use this HPTLC. Then fast identification of unexpected impurity during API production. So when there is a batch production was going on that time, some unexpected impurity you can detect by using the HPTLC, very rapid and simple technique. And further in by doing the HPTLC MS you can go for the identification of that unexpected impurity. Then content uniformity test is the best pharma application of the HPTLC. So very ideal for HPTLC to do CUT, that is content uniformity test. And typically CUT costs nearby 1,500 rupees by HPTLC, and up to the 8 CUTs you can do per day by using HPTLC. So double benefit, the time saving as well as the money saving. Next application is the botanical applications. First, you can see the identification of the whitest negundo, what is the chest tree leaf, by using the HPTLC fingerprint. So here, what we done, the agonoside and negondoside is the main marker component was applied, and the different species of these plants was developed with this marker. And at the same RF when they are coming, so on that basis we identify that it is present in the sample or not. Then identification of the ashwagandha, which is Withania somnifera by HPTLC. So you can see the discrimination of Withania somnifera root and leaf with withanolide A, beta cetosterol, and Withania somnifera leaf. So it is done and identify by using the HPTLC. Then identification of the kokum, which is Garcinia indica fruits by HPTLC fingerprint. So garcinol is the main marker component was present in the Garcinia indica sample that was detected by using the HPTLC system. Then identification of the nimesulide and diclofenac in Ayurvedic tablets. We know that the nimesulide and diclofenac is the synthetic APIs. It should be not present in Ayurvedic tablets, but sometimes some vendors is added there inside that manufacturers to give the rapid and fast results. So that is not allowed, and that was detected by using the HPTLC. Quantification of adulterant in xanthorrhizia or aromatica samples. So you can detect any adulterants in there in the sample. So by using HPTLC, you can detect it. Steroids in herbal formulations. Then by using HPTLC, you can go for the stability check studies. So there is a two methods. One is the quantification and one is the fingerprint method. So in quantification, you can quantify ki any degradation or anything is happen in the sample, or by using the fingerprint method, you can detect it if anything extra band was there, any one less band is was there. So according to that, you can do the stability check studies. Now some application is food. So USP chapter 202 for the fixed oil fingerprints. So here you can see that there is a 14 fixed oil samples was analyzed. So each one of them having their unique fingerprint. And if you compare that, or you can do this the analysis by using HPTLC, you'll get the same result for the oils. Then determination of aflatoxins B1, B2, G1, G2 in food stuff by HPTLC. As we know, the being fluorescent, most mycotoxins are easily detected by HPTLC in PPB and PPT level. So here we know dervatization was required, and if no other fluorescent substance is present, cleanup is not required. Then detection of sugars. Monosaccharides are very difficult to separate by any chromatographic technique, but HPTLC offer you very fast cost effective method for the separation and detection of sugars, and very simple and low cost. So here is the example of the glucose, sucrose, maltose and fructose, which was detected and identified after the derivatization. So here you require the derivatizing procedure. So aniline diphenylamine phosphoric acid ADPA was used as a derivatizing reagent for detection of sugars. Then nicotine in tobacco, gutka, mawa, and pan masala samples. So there is two images where it is visible in the UV before derivatization, but after derivatization it is showing the very prominent good orange color bands with the help of drag and drop reagent. Then detection and quantification of water soluble and insoluble dyes in food samples. So we all know Sudan dyes was banned because that it is carcinogenic. But if any manufacturer is adding in the sample, you can detect that by using the HPTLC. Adulteration of the toxic argaman oil in mustard oil by HPTLC. We know the sanguarine is a very toxic polycyclic ammonium ion present in argaman oil, or if any adulteration was done of argaman oil in mustard oil, we have detected that by using the HPTLC. Then detection of the palm oil adulteration in edible oils by HPTLC. We all know the palm oils is little bit cheaper as compared to the edible oils, and many times some of the manufacturers they added that palm oil in edible oil as adulteration. And if there is any adulteration, you can detect by using the HPTLC, and we done that analysis and we detected. So this is the example. Then identification and quantification of the MSG monosodium glutamate in seven food different samples like noodle, pasta, and raw products. We all know before some years there was a Maggie was banned because of the MSG. So it should be not present. But MSG as a flavor enhancer was added in the foods like noodles and pasta to give the good flavor, and that is also we can identify and quantify by using the HPTLC. Now, new application fields for instrumental and TLC, HPTLC. In forensics, they have specific inks for their secret service and government. So, if there is any doing any fudging was there ink, you can detect it by using the HPTLC. Now, these are the some books and literatures available on HPTLC for method development and validated methods. Like HPTLC Association was there. So you just go on Google and type HPTLC Association. You will get the 100 plus validated methods related to the herbal samples. You have to log in with your login ID and password. You register, and then you can use that method for your analysis. Then CAMAG website also you will get some of the application notes for different fields like pharma, herbal, forensics, food, and these are the some famous books for HPTLC. So as I told you for the HPTLC methods, so this is one screenshot of that method. So here you can see this type of method you will get where it give all the information how to prepare the sample, which standard you should use, which mobile face you should use, and how the bands will look like the pattern. So everything is there in that method. Then she is Dr. Vandana Gawande, madam. She won that Euro 2000 scholarship in Berlin. I shown you one example of the Cipexim and azithromycin degradation study. So she done that analysis, and for that research work, uh, she won that Euro 2000 scholarship. Then this is the gold prize winning poster at HPTLC Symposium at Bangkok in November 2018. So Yugendra Patil and research guide Sunita Shailaja from the Ruya College, they won this award. Then Ancrom philosophy, actually we are technologists, we are not traders. Ancrom wants to be your analytical partner, not merely a supplier. We will keep your Kamag HPTLC system productive for years due to our excellent service, laboratory support, and focus on the customer. Now, newly expanded Ancrom HPTLC lab in the Mumbai. Will give unlimited support to the customer anytime they want. HPTLC accessible to one and all Indians. All Mac software and instrument for training and analysis. And 14 analysts, including two PhDs, have in our lab. Separate set of instruments for hands-on training. We have PhD earn and learn scheme also there in lab, and lab is available for your publication works by using the HPTLC. We give HPTLC education webinars with the live demo. As I told you, we give the free training like for all government labs and postgraduate student. Training duration is flexible. We can decide the date mutually as per convenience. And the after training, we'll give the certificates also. Today also after the webinar, within the 1 week you will get the certificate for this webinar, everyone. And over 5,000 scientists was trained free since 1994. So this is our email ID and contact number. If you have any queries, doubt, you want to discuss anything related to the HPTLC, you can mail us or you can call us on this number. Ancrom aims to make India as a world leader in use of HPTLC. So our lab give access to all Indians who wish to use the HPTLC but they do not have. So thank you very much. So now I completed my presentation part. Now we'll start with the demo part. So my dear colleague uh, Vishwajit will show you the demo part. So I will stop to sharing my screen. Wait, Vishwajit. I will stop sharing my screen. Yes, yes, just a bit. Yeah,
Thank you, Mr. Ramesh
Vishwajit,
Kalande.
Vishwajit, just a minute. Yes, ma'am. Yes, ma'am.
Okay, now it was indeed a very organized and in detailed presentation. I am sure our students and other participants have benefited. We will take up the questions, but before that, I would like to request Dibakar sir to introduce our next speaker for the live session, Mr. Abhijit Khale.
Madam,
So, Dibakar sir, over to you.
1 minute, ma'am.
Yes,
Ma'am, Abhijit Khale was busy with some another important work, so we just
okay,
now change in the some demo part. The Vishwajit Khale will take part of the demo in this webinar.
okay,
Okay?
okay, fine.
Yeah.
Okay, sir, okay.
विश्वजीत वर्टिकल है हॉरिजॉन्टल करना स्क्रीन हाँ मैडम कैन वी स्टार्ट द डेमो
Yes, sir, you can proceed.
ओके 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 फाइन विश्वजीत ए स्क्रीन दिसत नाही नीट तो तू दिसत नाहीयेस नीट पकड कॅमेरा चेहरा दिसत नाही तू उचल बघा चेहऱ्यावर
Okay, Vishwajit sir, you are not audible. We cannot hear you. Your voice is not clear. No, sir, it is still not clear.
Sorry, we are facing some technical. Now I am audible, ma'am.
Yes, yes, now it is clear.
Amit, are you there in this session? Amit, am I audible?
Yeah, yes, Vishwajit, you are clearly audible. You can start the demo.
Okay. Ma'am, please check the connectivity at your site also because my colleague is saying
Yes, sir,
I'm
it is
audible.
audible.
So he
Now you're audible,
started
sir.
the
Yes.
session from another PC. Ma'am, now I'm audible and clearly visible.
Yes, sir, you're audible and visible, sir. You can proceed.
Okay, ma'am. Thank you. So I'm Vishwajit Kale from Amcrom R&D Center at Mulund, Mumbai, specially dedicated lab for HPTLC research and development and all CAMAG HPTLC instrumental modules you can see in this laboratory. I will show you the short demonstration from live from our laboratory from Mumbai. So along with me behind the camera, my colleague Mr. Siddhasari is there. He is holding the camera. I request Siddesh to come inside to show our laboratory. Yeah. So this is our R&D center. One lecture is going on in conference room. We have a separate conference room for training session for presentation and all. Right now, a student training program is going on. So this is our library for referencing and literature survey. Literature survey plays vital role during any type of analysis related to chromatography or spectroscopy. So we have different books available with us in different application fields for food analysis, herbal, and many more. We are referencing. We have all ICMR volumes for referencing in herbal research. Then pharmaceuticals analysis. For that, we are using Dr. P.D. Sethi, Sir book, etc. So this is our library. I request Siddesh to come inside. This is the entrance of our laboratory, CAMAG HPTLC R&D Center, dedicated technique, CAMAG HPTLC. Here you can see all the modules of CAMAG HPTLC. We have separate zones like for training, for innovation. Then in these sections, we are taking trainings as well as we are doing our day-to-day activities. हाँ हाँ उसने बताएगा रिपोर्ट में आएगा वो एसएसटी पहले एसएसटी पास हाँ मोबाइल डेटा चालू होता है
Hello, sir. Hello.
Hello. There is some technical issue related to the network. We'll resume in 1 2 minutes. Huh? Sorry for
Okay, okay,
inconvenience.
sure.
Yeah.
It's okay, sir.
Good afternoon again. We have digitalized the network connectivity. I hope I'm clearly audible.
Sir, you are not clear.
Vishwajit, no, video is not clear. Even the audio is also breaking.
Okay, okay. Now it is okay.
Yes, now it is fine.
Okay.
Okay,
So
video is not clear. Voice is clear
basically,
now, but
we are
video
seeing
is blur.
all the basics theoretical aspects of HPTLC during the presentation by my colleague Amit. So now we will see all the instrumental modules involved in HPTLC system.
Video blur है पूर्णपणे. Now
दुसऱ्या फोन मधून लॉग इन कर.
hello, madam. Okay.
Yes, sir.
We joined from the different device for the our demo part. Okay, Anu?
Okay,
Mm,
okay.
yeah.
Now I'm visible on screen, Amit.
No, Vishwajit. Just now, just now. Okay.
Clear?
Okay, you can start. Yeah.
Okay, my audio is clear and video is clear, Amit.
Yes,
Yes,
sir.
yes,
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 yes.
yes, Vishwajit.
Okay, yeah, yeah. So this is the area where we are keeping all the CAMAC HPTLC modules in our R&D center. Here we are only dealing with CAMAC HPTLC and related system modules. Yeah. So this is the stationary phase involved in HPTLC analysis. There are more than 50 different types of stationary phases are involved. We can choose as per the analyte of interest in our hand. We can choose any suitable stationary phase and we can start with HPTLC analysis. To perform each step involved in HPTLC analysis, there is a separate module in CAMAC HPTLC system. Now we will see one by one all the modules, important modules involved in the system. So this is the stationary phase. This is 20 by 10 centimeter dimension TNC silica gel plate. Pre-coated plates mostly we are using for day-to-day analysis, manufactured by different manufacturers. As per number of samples we want to analyze and number of standards, we can select a suitable dimension of the plate. The system is compatible for 20 by 20 centimeter or less than that, any dimension of the plate we can select as per our need. After selection of a suitable stationary phase, we need to apply the samples and standards which are in liquid form onto the plate by using HPTLC applicators. So this is a CAMAC ATS4 automatic TLC sampler HPTLC applicator. Here we can keep at a time 66 samples or standards in the vial rack. Auto sampler tray is there, and there is a syringe, mixed volume Hamilton syringe inside, 25 microliters or 100 microliters. We can apply 1 microliter to 100 microliters onto the plate as per our need. So it is more and more accurate sample application without touching the stationary phase. So this is contactless application and spray on technique. Nitrogen gas is connected or compressed air is connected to generate a fine spray of liquid sample. That spray will fall onto the plate, and we are applying samples and centers in the form of bands and not spots. So band-wise sample application will give us a more good resolution after chromatographic development. This is the use of CAMAG ATS4. At a time, 66 samples we can keep. A syringe rinsing and filling is completely automatic, and we can connect this ATS4 module to software, Vision Cut software, and we can make a sequence where we want to apply the bands, how much volume we want to apply, etc. Onto the plate that we can set into the software. And on the stage of this ATS4, we can keep the plate that is stationary phase. After completion of sample application, the next step involved in the HPTLC analysis is chromatographic development. For that, we are using different modules. That is manual development module, that is twin interrupt chamber, or ADC2 automatic development chamber, or AMD2 for gradient type of HPTLC plate development we can use. After chromatographic development, the next step is photo documentation. For photo documentation, we are using CAMAC TLC Visualizer 2. This Visualizer 2 is fitted with highly sensitive industrial grade camera and three lamps to generate three different illuminations: deuterium, mercury, and tungsten, to generate a fluorescent light as well as shorter wavelength, longer wavelength UV light, and white light, 540 nanometer white light. So we can capture the plate images automatically through software in this CAMAC TLC Visualizer 2. Just we need to keep the plate after chromatographic development into the visualizer and give command from the software. This is IQ/OQ compliant in CGLP environment 21 CFR Rule 11 compliance environment. We can use this TLC visualizer. Many reputed pharma companies across India install this TLC visualizer in their laboratories to capture the plate images after chromatographic development. So photo documentation and image of the separated band is the key fact in HPTLC analysis. Visual observations plays vital role in HPTLC analysis. After that, the next step is quantification and identification by using spectra. So we can do quantification as well as spectral analysis in CAMAC Scanner 4. This CAMAC Scanner 4 is acting as a detector in HPTLC system, fitted again with three different lamps: deuterium, mercury, and tungsten. By using grating as a monochromator, we can generate 190 to 900 nanometer any single wavelength as per the lambda max value of our compound of interest. Just open the shutter of this scanner 4, press load button, the stage of scanner 4 will come out, and on the stage we can keep the plate for scanning densitometry. And again press load button, plate will go inside, close the shutter. All the things we can control through software. In software, we need to put which lamp we want to illuminate, which wavelength we want to generate to scan the plate, in which range we want to capture the absorption spectra. So basically, it will generate a chromatogram, absorption versus RF value response. Based on the peak area and height, we can quantify this particular substance, and then we can proceed for spectral identification. We can generate absorption spectra of any separated band onto the plate in the range of 190 nanometer to 900 nanometer by using this CAMAC Scanner 4. So this is very much sensitive. For fluorescent absorbing compounds, we can go up to picogram level. For UV and visible sensitive compounds, we can go up to nanogram level of detection by using CAMAC TLC scanner. So I request Siddesh to come with me. I'll show you some other modules, important modules in HPTLC system. This is CAMAG ADC2. This is CAMAG ADC2 completely automatic chromatographic development chamber. Here, normal twin trap chamber we are placing, and the upper assembly will completely enclose the chamber. And in this upper part, we can hang our plate of interest just like this. So this plate is in my hand. So I am hanging this plate on the upper part of the ADC2. Just hang the plate 20 by 10 centimeter or less than that dimension. Pour in the mobile phase, that is 10 milliliter for chromatographic development and 25 milliliter for chamber saturation. And this is again software control with humidity control option. So after giving command from software, plate will go automatically into the chamber, into the mobile phase, and after chromatographic development, plate will come out from the chamber in the upper part. Plate drying is also automatic. The pump is given to dry the plate. We can decide the plate drying time 5 minutes or 10 minutes as per the convenience, as per our convenience and method requirement. So the main feature of this ADC2 is humidity control. We can control the humidity of the plate during chromatographic development. We can control exact 33% humidity by using MgCl2 hexahydrate saturated salt solution. Or if we fill this jar with any other salt solution or like molecular sieves, by using molecular sieves, we can maintain 0%, 0 to 5% humidity of this chamber during chromatographic development. Humidity plays vital role in separation of the bands during chromatographic development by using mobile phase. The next module is automated multiple development module, that is CAMAG AMD2 module. Here, this we are using for gradient type of HPTLC. At a time, we can place 1, 2, 3, 4, and 5 different solvents or 5 different mobile phases in these jars. And in this assembly, we can hang the plate here 20 by 10 or less than that dimension. And the gradient program we can select into the software. The gradient program is based on the polarity of your applied sample or applied analyte and polarity of different solvents you have kept into these jars. So mostly in column chromatography, we are changing the gradient program as per RT value. But here we can change the gradient program as per distance traveled by solvent. It will be polar to non-polar. We can separate all the applied components at application position at different RF values by setting up a proper gradient. I hope you have seen that application separation of different lipids by using AMD2. So this was about automated multiple development module in CAMAG HPTLC system. So the another one module is manual type of chromatographic development module, that is CAMAG twin trap chambers. So there are three different dimensions of this chamber, that is 20 by 10 centimeter, 10 by 10 centimeter, or 20 by 20 centimeter. As per the plate dimension, we can select any suitable module for the analysis. So we are using saturated chambers. Saturation is by using Wattman number 1 filter paper. We are keeping at one side. We are pouring the mobile phases. We are tilting the chamber at 45 degree so that mobile phase will distribute itself between two troughs in equal volume. And then we are keeping the plate in the rear trough of the chamber. And by capillary action, mobile phase will move and separate out the components. So here, this is our wet laboratory area. You can see another device, another module by CAMAG, that is CAMAG Immersion Device 3, to dip the plate into the derivatizing reagent. Now, some molecules are not absorbing any wavelength of light and very difficult to detect on plate. So how to detect that molecules? We need to do derivatization in that case. There are many types of derivatization process, but major two types are dipping the plate into the reagent or spraying the reagent onto the plate. So for that we have CAMAG immersion device for dipping the plate into the reagent. On this hanger we can hang the plate I will show you demonstration how we are doing it. So this is 20 by 10 plate. I kept this plate on holder. I'm just keeping this holder here on emergent device, and after pressing this red button, the holder will go automatically downwards, and plate will get dipped into the derivatizing reagent, and within seconds it will come out automatically. That dipping time we can set by using this knob. For 1 second, 2 seconds, we can keep the plate dipped inside the derivatizing reagent. After dipping the plate into reagent, the next step is uh, plate heating. Without plate heating, reaction between reagent and molecule will not get complete. So for that, we are using CAMAC TLC plate heater 3 module. Here on this module, we can control exact 25 degrees Celsius to 200 degrees Celsius, any single temperature. So the surface of this TLC plate heater is very much robust and regular towards acid or bases which we are using in preparation of derivatizing reagents. Then temperature sensors of this TLC heater, plate heater, are very much sensitive and very much capable to generate very uniform temperature throughout the surface. So therefore, plate heater plays a vital role in very uniform derivatization. And after derivatization, we can do even quantification very accurately and precisely. And another module for derivatization is CAMAC derivatizer. To generate very fine mist of derivatizing reagents, we can use this. That very fine mist will fall down, settle down onto the stage of this derivatizer on which we have kept our plate. So take the stage out from the hood, keep the plate and insert the stage inside. Take the hood of this derivatizer downwards, and there are 4 different color coded nozzles: blue, red, yellow, and green. As per the viscosity and properties of derivatizing reagent, we can select a suitable nozzle. In this nozzle, just pour in 2 milliliters of derivatizing reagent. By piezoelectric action, this nozzle will generate very fine mist, aerosols of the